All right, boys and girls, need you to bear with me on this lecture. This lecture is going to be a little more involved, a little longer than some of the previous ones. This one's going to be dealing with proteins, and there's a lot going on with the proteins. So, let's sit back, take your notes, follow along with your handouts, please. Now, when it comes to proteins, proteins are going to be pretty much our most abundant organic molecule. Proteins are responsible for all the work done in your body. Yes, a lot of people want to think, oh, it's the tissues, oh, it's the cells. Tissues are made of cells. Cells are just bags made by phospholipid membranes. They don't do anything. It's the proteins inside them that regulate, build, make, break down, replace. They act as a structure. They give the cells their structure. A nerve cell looks different than a skin cell, looks different than a muscle cell. And that's all because of the proteins present that are giving it its structure. They're protective. They allow for transport, the movement of things in and out of cells. And once in the cell, movement around the cell is done by proteins. And as I've said in a previous lecture, enzymes. Enzymes are what make our metabolisms go. Yeah, glucose would break down, release its energy, given time. Days, weeks, months, years. Our cells, our bodies can't wait that long. We need things to happen in seconds, milliseconds. That's what enzymes do. They cut, paste, twist, tweak, whatever. So let's look at enzymes. Let's talk about these guys. Upstairs in chemistry hell, they like to use the term catalyst. A catalyst is something you add to a reaction mix to trigger the reaction. Upstairs in chemistry, this can be a metal, this could be some compound, this could be something there. Well, in a biological standpoint, we can't have metals and things like that just willy-nilly being added in, because we can't control it. And biology is all about controlling how things go. That's what your metabolism's all about. So, along comes enzymes. Enzymes are a protein catalyst. The reaction's going to occur, it's just going to take time on its own. Remember, a couple of lectures back, the first one for this chapter, talked about sucrose. You take a bunch of sucrose, sugar crystals, dump them in water. You wait days, weeks, months, year. All that sugar will break down into individual components, glucose and fructose, but it's going to take time. An enzyme acts as a catalyst. It'll make that reaction happen, and it'll happen in a time frame that is biologically useful. Minutes, seconds, milliseconds, as opposed to days, weeks, months, years. Now, I've said previously that enzymes, my favorite saying for enzymes, they twist, cut, paste. They do one thing and they do one thing only. We find enzymes fall into three categories: catabolic, anabolic, catalytic. Catabolic, take substrates, break them down, go through, cut it up, cut. Take something, a big polymer, cut it down into the monomers. Our hydrolysis reactions from previous lectures. Anabolic, take the monomers, string them together, dehydration reactions. And then there's catalytic. Cut, paste, twist, tweak to take a substrate, turn it into a product, as they're showing here. This enzyme, maltase, takes malt the sugar, maltose, which you can see here, 1, 2 sugars, so it's a disaccharide. Fits in the middle This ribbon you see here is to give you a three-dimensional representation of the enzyme. The substrate fits right into special place within the enzyme, and the catalytic effect of it. Cut, paste, twist, tweaks, something. Pick only one. Here, this one cuts. It cuts maltose, releasing two glucoses. That's all it does. As long as there's maltose present, it'll keep cutting. Cut, 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 cut. It will do nothing else. The enzyme has the specific shape to do this one and only one thing. You find the different proteins have different functions throughout the body. Transport, structural, hormones, defense, immunoglobulins, your antibodies. No matter what the protein is, has a specific monomer, the amino acid. All amino acids have a theme in how they are formed, in their form, in their shape. You have what is referred to as an alpha carbon. This is a central carbon that everything is built upon. Remember, organic molecules, carbon. To the left, you have the amino group. Remember, this is one of our functional groups. Amino, because it has a nitrogen, and carboxyl. It's called an amino acid because here's your amino group, and the carboxyl will lose that hydrogen, making it more acidic, hence amino acid. Notice this R right here. This R, when you see an R like this in any type of structure that's been drawn, it means insert here. Insert what? Well, depends. Turns out the amino acids that make up all the proteins, all the polypeptides in your body, fall in one of these groups. Nonpolar, polar, positive, negative charge, or a ring. Notice, alpha carbon, carboxyl, amino. And in the blue that you see in every amino acid, that's the R group. The R group can be anything from just a hydrogen to large cyclic rings and everything in between. Notice N plus COO negative. So the amino acid itself, if it's individual, does have a slight charge, but this nonpolar, polar charge thing comes about more to play when it's linked together, forming a polypeptide chain. Then this N plus, the COO minus, gets taken up in the polypeptide, the chain itself. Now, writing out valine, writing out aspartic acid all the time gets to be a bit much. So, early biologists who were studying this, figuring out the different amino acids and how all this works, came up with two means of nomenclature. A singular letter or a three-letter designation. Valine, V or val, aspartic acid, D or ASP. Now, V for valine makes sense. D for aspartic acid, say what? Well, it depends on what order they were found. They went A, B, C, D, E, F, the alphabet, and here you go. Now, you've probably heard the term essential amino acids. What are the essential amino acids? Well, these are amino acids we can't make. These are amino acids that, well, we have to take in our diets, whether it's from a plant or an animal that ate a plant. We need isoleucine, leucine, and cytosine. Now, what's interesting about all of these amino acids is when you string them together, they automatically start forming three-dimensional shapes. The three-dimensional shape, as we saw in the previous slide with maltase, cutting maltose, that overall shape gives it a function, a specific function. And it all ties back to what's the sequence of amino acids. Now, glycosidic ester, now we have peptide. Peptide bond. It's a dehydration reaction giving us a carbon-nitrogen bond between two neighboring amino acids. 3 groups so far, 3 different types of bonds. Keep up with them. Now, so far I've used the term polypeptide, I've used the term protein. Turns out these things are 2 different things. You take a chain of amino acids, one long chain of amino acids. That in and of itself is what we refer to as a polypeptide. That polypeptide will fold in on itself in specific ways, all the R groups interacting and things like that, to give it a 3-dimensional shape. Now, difference between a polypeptide and a protein. Protein is one or more polypeptides that have the specific correct shape to have a function. A singular polypeptide on itself may not have a function. It may need to be bound to other polypeptides to actually form something that works. Hemoglobin is a great example of this. Hemoglobin is composed of 2 alpha polypeptides and 2 beta polypeptides. Individually, an alpha, individually a beta, do absolutely nothing. But those 4 polypeptides coming together to form the complete protein, hemoglobin, well, that actually works. That actually does something. Now, how do these polypeptides get formed together? Yes, there's the peptide link. But it turns out this is something that's going to occur during what is referred to as translation. Translation is ribosomes stringing together. Stringing together what is referred to as the primary structure of a polypeptide. Turns out there's 4 levels of structure. The simplistic, the primary, the advanced 3-dimensional shape, which is the quaternary. The primary structure is literally nothing more than the string of amino acids. Amino acid 1, 2, 3, 4, on through the hundreds of amino acids that are going to make up that polypeptide chain. There you go. That's primary. The primary sequence is based on your genes. Remember in the previous lecture I said your DNA is instructions on how to make and maintain you. Well, there's the instructions. How to make a protein. What amino acids to link together in the primary structure. That's what a gene is. That's all. Sometimes, though, weird things happen. There is a genetic disorder referred to as sickle cell anemia. Sickle cell anemia primarily affects people of African descent and some of Middle Eastern. What happens is the gene gets mutated. The instruction gets altered. So that instead of glutamic acid, you end up with a valine. Oh no, doesn't sound like a big change, but look at the shape. Look at the R groups. The R groups are completely different. The primary structure dictates the secondary. Secondary dictates tertiary. Tertiary dictates quaternary. You change the primary, you change everything else. Turns out that this is part of the beta chain of hemoglobin that I just talked about. 2 beta chains, 2 alpha chains come together to form hemoglobin. Under normal conditions, hemoglobin is free floating throughout your red blood cells, has the ions in it that's going to pick up molecular oxygen, deliver it throughout your body. unless there's this change. Instead of glutamic acid, you now have valine present. Instead of nice three-dimensional shape beta chain, beta subunit, you now get this beta subunit that's constricted. Still forms two betas, two alphas, but instead of the hemoglobin free floating, it now gloms together, forms long crystals that distort the shape of the red blood cell. You've seen pictures of red blood cell. They're nice, they're round, they got a little dimple, they look like a donut. Free floats throughout your blood cells, throughout your blood vessels, sorry. Except in sickle cell anemia. The hemoglobin globuling together, forming these crystals, distorts the shape, stretches it out, distorts it until literally it looks like a crescent moon. Turns out these crescent moons get caught up in capillaries. People with sickle cell anemia will spontaneously start having bruises, bleeding. Because the hemoglobin's not shaped correctly, it doesn't function correctly. Means that less oxygen is carried throughout the body. People with sickle cell anemia don't live as long. Their metabolic rates are much lower. They have issues with development, physical as well as mental, because of the reduced blood oxygen levels. And it's all because of a change of one amino acid. You look here, you can see some of the red blood cells look somewhat normal. Some of them, as you see right here, have that sickle cell shape. That gets caught up, causes blood clots, stroke, causes bleeding, internal hemorrhaging, bruising. So, primary structure is the string of amino acids based on what the genes say to link first, second, third, fourth, on and so on amino acids. Well, these amino acids start to interact with each other in their chain, in their polypeptide. They push, they pull against each other. And they can form one of two three-dimensional shapes. An alpha helix, think corkscrew, or what is referred to as a beta pleated sheet. Think like a flat plane that's got some ridges in it. A corkscrew or an up and down, almost like a folded map, unfolded map. Corkscrew and the ribbons here are to show that this is a beta sheet. Two completely different structures, which means different shape, different functions. So, primary, string of amino acids, the polypeptide, pushes and pulls, forms either a helix or a sheet. Now, the sequence of helices, the corkscrews, or sheets, is what tertiary is all about. Tertiary, now you have the R groups, pushing and pulling against each other, making covalent bonds, making hydrogen or ionic bonds, holding things together. You have all of the different bonds we've talked about in Chapter 2 coming into play. Disulfide bridges, linkages here, hold things together. It takes energy to form it, means it's going to take energy, some sort of an enzyme to break it. Ionic bonds, positive, negative, interacting, pulling towards each other. So what you could have here is a sequence of helices and sheets. Primary is a sequence of amino acids. Secondary, does that sequence form a helix, a corkscrew, or a sheet? Tertiary, what's the sequence of corkscrews and sheets? Which leads us to quaternary. Is that single polypeptide a functioning protein, or does it require more than one polypeptide to have a function? So when you look at it, string of amino acids, helix, sheet, what's the sequences of helixes and sheets, and then how many polypeptides does it take to get a complete protein. Now, once proteins are formed, doesn't mean they're permanent. They fall apart. Takes energy to make the bonds, takes energy to break the bonds. Well, sometimes that energy comes in the form of heat. Heat is energy. Heat is thermal energy. Add enough heat, ionic bonds, hydrogen bonds fall apart. Add even more, the disulfide bonds break. You lose quaternary, you lose tertiary, you lose secondary. You now just have a chain of amino acids, a polypeptide. Look at the egg there on the right. Denaturation is what they call it. You're cooking the protein. You're making it lose quaternary, then tertiary, then secondary structure. All that's going to be left is primary. Denaturation. This can be due to temperature, if you're cooking, but also pH. Remember, ionic and hydrogen bonds are holding some of that tertiary structure together, holding the quaternary structure together. You add in a bunch of free hydrogens, lower the pH, because remember, pH is nothing more than a logarithmic measure of how much hydrogens are free in a solution. More hydrogens free, lower the pH, the more acidic. So you change the pH, you lower the pH, you denature proteins. The proteins begin to fall apart, unravel.